Science Research Center from 1999 to 2012. He was the recipient of the UAB President's Award for Excellence in Teaching and was also named an Association for Research in Vision and Ophthalmology Silver Fellow in 2009. He served as chair of the Council of Center Directors in 2007 and currently serves as the chair of the Research Advisory Group. He received the Sam Brown Bridge Builder Award in 2011 and was named University Professor, one of only 12 in the history of UAB in 2013. I would like an explanation of that. He currently also serves as the Director of Research Commons of the Center for Clinical and Translational Science. Dr. Kyser's research and teaching activities have been funded by the NIH, NSF, and several private foundations. Dr. Kyser was appointed as Assistant Vice President for Research in January 2013, and then as Associate Vice President for Research in 2015. Among other responsibilities, he oversees programs to identify and promote applications for extramural research funding, is also developing and promoting an alternative process for funding major research equipment maintenance and replacement. Please welcome Dr. Kyser.
Well, thank you very much. I'm going to go ahead and try to share my screen. Let's see if this works this time. Is everybody seeing the full screen? Okay, so we're good. Okay, well, I'm really grateful for the invitation to speak with you today. When Janice first invited me, the topic was supposed to be research at UAB. And when I thought about it, I realized that with the total research budget in 2019 of well over $600 million, to try to talk about UAB research, I'd be able to barely scratch the surface in an hour. So then last week, I think I got another email from Janice saying that she was looking forward to hearing about UAB research, especially that related to COVID-19. So this talk now is COVID-19 research at UAB, a much more targeted topic, and one that we have quite a lot to say about in terms of UAB's role in trying to affect the pandemic. Okay, what slide are you seeing now? The same one? Okay, so this building, can you see my pointer? No, I guess not. Okay, can you see my pointer now? The green thing?
We could see it before, or is it?
You could, okay.
When you first asked, it was up there.
When it's up here, can you see it?
There, you got it.
Okay.
We can see it.
This is the building I work in when I'm on campus, which I haven't been very much for the past 10 months. That's the administration building. Just to get you oriented, this is Children's Hospital over here. This is Radiation Oncology and the Women and Infant Center. The VA is right in here, just so you're oriented to the campus. Okay, so I thought what I'd do first is give a brief history of the coronavirus. You're seeing that slide? Is that okay? Good. Okay, just to put this in context, beginning in December 2019, patients with pneumonia of unknown etiology were identified in the city of Wuhan, China, a city of about 11 million people. The causal agent, which turned out to be a new type of coronavirus, was identified around January 7th in 2020. And as of the 21st, there were more than 270, probably more than 300 cases confirmed in Wuhan. On January 23rd, 2020, Wuhan city was locked down by the local government. And on January 30th, the World Health Organization declared a public health emergency of international concern. The first laboratory confirmed case of COVID-19 in the US was recorded on January 20th, 2020, and reported to the US Center for Disease Control on January 22nd. On February 11th, 2020, the International Committee on the Taxonomy of Viruses announced severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2, as the name of the new virus. On March 11th, the World Health Organization made the determination that the COVID-19 was a pandemic. On March 16th, UAB moved to a limited business operations mode, and this was done to help control the spread of the illness and protect faculty, staff, students, and patients. We were in the limited business operations mode through the spring and into the early summer of 2020. The pandemic worsened very quickly over the next 8 to 9 months. As of yesterday, more than 96 million cases have been diagnosed worldwide, with more than 24 million cases diagnosed in the US, and more than 4,000 people in the US have died. In Alabama, as of yesterday, there were nearly 430,000 cases, with more than 6,000 deaths. And just to make this very clear, we have never seen anything like this virus. As bad as these numbers sound, the numbers of cases and deaths are probably the actual numbers are probably very much higher. The undercounts are attributable to lack of testing and lack of adequate diagnostic capacity. Now I'm going to ask you to think back nearly 20 years to the terrorist attacks of 9/11. 7 days after those attacks, letters laced with anthrax spores began appearing in the US mail. An unknown number of people received these letters. We know of at least 7 people who received the letters. So anthrax is a disease caused by a spore-forming bacterium. It mainly affects animals, but humans can become infected through contact with animals or by inhaling spores. The symptoms in people depend on the root of the infection. Inhaled anthrax is really, really hard to treat and can be fatal. 5 Americans were killed and 17 were sickened in what is the worst bioterrorism attack in US history. The ensuing investigation by the FBI and its partners, which was codenamed Amerithrax, is one of the largest and most complex investigations in the history of law enforcement. And at the end, when we have questions and answers, I'm happy to come back to this and talk more about it if you'd like. So the anthrax attack raised the specter of further bioweapons attacks on the United States. As a result, the federal government awarded 12 contracts to build what were called regional biocontainment laboratories across the country. These were designed to support research capabilities that would be needed to develop drugs, diagnostics, and vaccines for emerging infections or bioterrorism attacks, and to provide surge capacity in the event of a public health emergency. UAB is part of a consortium that was funded by the National Institutes of Health known as the Southeastern Regional Center of Excellence for Emerging Infections and Biodefense. In one of the regional biocontainment labs, funds were awarded to UAB for the construction of one of those. It was finally completed in 2008. And it provides secure laboratory space for research involving dangerous infectious agents such as SARS-CoV-2, the virus that causes COVID-19, the disease COVID-19. So the research in this highly secure building is really ramped up over the past 10 months. It includes actually producing SARS-CoV-2 virus for other scientists to use in their experiments. And these experiments span from the effect on lung tissue, the effect on other organs. And one set of studies has been focused on finding an animal model that accurately recapitulates the conditions seen in humans when they're infected with SARS-CoV-2. Animals are really important because they can be used to better understand the virus and its effects on the body, to test new treatments. Often mice are used as an animal model for human disease, but in this case, for COVID-19, they don't exhibit many of the symptoms observed in humans. And the symptoms they do show are much, much less severe than in people. So finally, efforts around the world have identified a good animal model for COVID-19, and this animal is the Syrian hamster. We can talk about more about that later in the session too, if you'd like. So given this backdrop, in the late spring and summer of 2020, local business leaders and other donors raised over $1 million to fund grants from UAB investigators that have the potential to have an impact on the COVID-19 pandemic. There were a total of 2 rounds of funding, and the total number of applications approached 100. We were only able to fund a total of 24 because of the lack of additional funding. Just to give you an idea of the type of research that was funded, the titles of some of these projects are glucocorticoid treatment of COVID-19 cytokine storm syndrome, individual and area level risk factors for COVID-19 disparities in the Deep South. And I'll have more to say about disparities later in the talk. Therapeutics targeting COVID-19 entry into pulmonary epithelial cells. Molecular mechanisms underlying the pathogenesis of acute respiratory distress syndrome in critically ill SARS-CoV-2 infected patients. So a wide variety of topics, and the first round of those awards were funded for 6 months, and those studies have now been completed. And many of those allowed UAB scientists to collect preliminary data that led to successful applications for follow-on grants from the federal government. And these follow-on awards are funding further development of new methods of early detection or more effective treatments. Also, some funds have been awarded for studies that will help us better understand how the disease can cause damage to so many different organ systems. Okay, I want to talk now about the role of pre-existing conditions in this pandemic. Now, according to the Center for Disease Control and Prevention, those suffering from certain chronic conditions are at much higher risk for severe COVID-19 disease. Among the most problematic conditions are obesity and diabetes. And we in the Deep South are in the bullseye nationally for diabetes. So this is a map, a heat map, essentially showing the prevalence of diabetes in the country, beginning in 2004, then in 2008, and then in 2016. The dark color shows the areas of highest prevalence. And the numbers below show that the darkest numbers refer to 12 to 33% of the population is affected. So we have this huge prevalence of diabetes and metabolic syndrome here in the Deep South. And this increased risk in our region is not limited to obesity and diabetes. Other conditions include heart disease, asthma, and other pulmonary problems, hypertension, and all of these also increase the risk for severe disease and death from COVID-19. Now, the national distribution maps for heart disease, hypertension, chronic obstructive pulmonary disease, asthma, stroke, all of these conditions would show very much the same national distribution that is shown here for diabetes. The populations in our regions suffer from a very, very heavy burden of chronic disease. And all these chronic diseases predispose people in our region for severe COVID-19 disease. Now, I want to stick on the topic of COVID-19 and diabetes for just a second. So a drug called metformin is often used alone or with other medications, including insulin, to treat type 2 diabetes. Now, this is a condition in which the body does not use insulin normally and therefore cannot control the amount of sugar in the blood. Metformin helps control the amount of sugar in your blood and decreases the amount of sugar you absorb from your food and the amount of sugar made by your liver. Metformin also increases your body's response to insulin, which is a substance made by cells in the pancreas that typically control the amount of sugar in the blood. So metformin is not used to treat type 1 diabetes. This is a condition in which the body does not produce any insulin and therefore can't control the amount of sugar in the blood. So just this month, in January 2021, one of UAB's leading diabetes research published a study that showed that patients using metformin before a diagnosis of COVID-19 experienced a threefold decrease in mortality when those patients were subsequently infected with COVID-19. So this is a huge decrease in mortality. This biological effect remained even after correcting for age, sex, race, obesity, and hypertension, or chronic disease, kidney disease, or heart failure. And the person who ran this study is Dr. Remdesivir, who is the director of UAB's Comprehensive Diabetes Center. However, how metformin produces this protective effect is not at all understood. Needless to say, there is significant ongoing research right now to try to understand how metformin provides this protection. One possibility is that metformin has a well-known anti-inflammatory effect. And it may be that this anti-inflammatory effect is the key to this protection in COVID-19 disease. Now, I'm going to move here from protection against disease to treatments for those that have contracted COVID-19 disease. So you may have heard of I'm sure you've all heard of the experimental drug Remdesivir. It was originally developed for treating hepatitis C and respiratory syncytial virus. It was developed through research conducted through something called the Antiviral Drug Discovery and Development Center, AB3C, which is centered at the University at UAB. UAB was awarded a $37 million 5-year grant from the National Institute of Allergy and Infectious Diseases. And the leader of this effort was UAB's Rich Whitley, who's in the Department of Pediatrics. So Remdesivir has been approved for emergency use among hospitalized COVID-19 patients. It directly targets an enzyme that the virus uses to continue to make more copies of itself. This is called an RNA polymerase. Remdesivir works by posing as a legitimate chemical building block for RNA production. The drug gets stitched into the nascent viral RNA strand and blocks further synthesis of the strand. The polymerase stalls out or produces a defective product. But Remdesivir is not perfect. It can't stop the production of all RNA viruses. So some viable copies of the virus continue to be produced. Nevertheless, in a number of clinical trials, it's been shown that patients suffering from COVID-19 who received Remdesivir were quicker to recover. And that, in this case, is defined as being medically stable enough to be discharged from the hospital. The median recovery time was 10 days with Remdesivir, compared to 15 days without Remdesivir in the placebo group. Remdesivir also improved mortality rates for those receiving supplemental oxygen. That is, the mortality rate is 4% of those receiving oxygen who are treated with Remdesivir versus 13% with a placebo at day 29 of treatment. The study also suggests that Remdesivir treatment may prevent patients from progressing to more severe respiratory disease. And it benefits patients mostly who are receiving supplemental oxygen. Now you notice I'm being very careful in how I report these clinical results because clinical studies are hard, they're complicated to do correctly, and I don't want to overstate the benefit of some of these treatments. Now, a number of other treatments have been tried and found to be efficacious in patients who are suffering COVID-19 disease. So corticosteroids, and that includes dexamethasone, prednisone, and related steroids, can be very helpful. And in one recent study, patients who required supplemental oxygen that were treated with dexamethasone were less likely to die within 28 days than those who received standard of care. Now, for this category of drugs, it's important to note that treating with steroids early in the disease can hamper the body's own immune response. And that's not what you want to do. So timing of the dosing with corticosteroids and the dosage itself are key. You may have also heard of monoclonal antibodies. So these are synthetically produced versions of the antibodies our bodies naturally make against the SARS-CoV-2 virus. These have been shown to be helpful in reducing mortality. Some of these were given emergency use authorization by the FDA in November of 2020, and they've shown to be helpful. but they're not currently allowed for use in hospitalized patients or those receiving oxygen therapy. And that's for a variety of reasons we can talk about later, but there's some indication that the monoclonal antibodies can lead to so an overly robust response. You've also heard, I'm sure, of convalescent plasma. So if you can recover plasma from recently recovered patients, there's patients who recovered on their own from the SARS-CoV-2, this plasma contains antibodies that their bodies produce to fight off the virus. A study published just this month reported that patients over 65 treated with convalescent plasma within 3 days, that is very early in the disease process, were 48 less likely to develop severe COVID-19 illness compared to placebo-treated patients. So this is huge. This is a major, major advance in our therapeutics for COVID-19 disease. But the very best way to avoid severe illness and treatment and require treatment is to avoid infection to begin with. As you've all heard ad nauseum, mask wearing, physical distancing, hand washing, all of those things are keys to avoiding infection. And now recently we have vaccines, although we don't have enough of them yet. And I want to take a minute just to talk about vaccines and the number that are in development and the 2 that are widely available for treatment. So there are well over 100 vaccines in development. I've given a timeline here. Let me just try to point here. Can you see this? Okay. So as of earlier this month, there were 41 in early safety stage trials. There were 22 in more widespread, that is larger safety and dosing trials. 20 were in widespread use for efficacy, that is how effective they are against the virus. 8 are approved for large-scale efficacy trials, and 2 are currently approved for wide-scale use. 1 had severe side effects and is no longer under development. Now, the 2 vaccines you've heard the most about and have been approved for use in the US are those made by Pfizer-BioNTech and another one by Moderna. And these are both types of vaccines that are brand new, called mRNA viruses. So I wanted to take a minute to explain those. And I hope many of you probably all already know this. Just as a general comment, I wasn't sure how many of you had biomedical background and how many of you didn't. So I tried to target this at an intermediate level that I think an intelligent audience such as this one probably can understand. Okay, so here's a cartoon of the coronavirus. It's a small particle. It has all these little spikes sticking off. So it has this corona or spikes, which is hence the name coronavirus. So SARS-CoV-2 is closely related to the first SARS virus, which you may remember originated in China and was associated with civet cats. There was another related virus called the Middle Eastern Respiratory Virus, syncytial virus, MERS, which occurred a few years ago as well. SARS-CoV-2 is related. So to make the virus, scientists generated what's called messenger RNA that encodes a sequence of the spike protein. And this sequence of the spike protein is the part of the spike protein that allows it to bind to molecules in our respiratory tracts. It can begin the initiation of the infection. So scientists can make artificial mRNA sequences that code for the SARS spike protein.
Yeah, ready now. What time do you think we should do?
The mRNA sequences are encoded in lipid nanoparticles. And these are what make up the vaccine. So the vaccine is injected into your muscle. And once it arrives, the cells take up the nanoparticle encoded mRNA sequences. And then those mRNA sequences are used in your own cells to synthesize spike protein. Those spike proteins are inserted into the surfaces of your cells, and your own immune system recognizes the spike protein as a foreign particle and makes antibodies against it. Now, these antibodies, the way these antibodies work is that they block the binding of the virus to your lung cells, to the proteins in your lung cells that the coronavirus can bind to. So your antibodies can block that binding and thus prevent disease. Now, the two viruses that have been approved for use, the Pfizer BioNTech and the Moderna, have been shown to be better than 94% effective. This is unheard of. We've never had vaccines that have this degree of efficacy. Typically, vaccines are in the 70 to 90% effective. So this is remarkable. And it's this new technique for making vaccines shows enormous promise for things like Ebola and other diseases that could become pandemic. Now, even after vaccination, some people may still have reservoirs of active virus, for example, in their nasal passages, even though they show no symptoms. So mask wearing, even after vaccination, should be still strongly encouraged. And for those of you who have been vaccinated already, please, when you're out and about, wear your mask. I know it seems cumbersome and after you've been through the vaccination process, but it's really important because you could still be a source of live virus. I want to talk about, for a minute, vaccines, public opinion, and disparities, health disparities. So nearly half of Americans say that vaccine distribution should be the new president's top priority. A little over 60% say they will be vaccinated when adequate supplies are available. So this 60% is a real problem. The National Institutes of Health has responded with something called the Community Engagement Alliance against COVID disparities. They shorten that to CEAL. This focuses on addressing misinformation around COVID-19. It seeks to engage trusted partners and messengers to deliver accurate information and to educate communities on the importance of inclusion of these communities in clinical research to overcome 19 and research in overcoming health disparities. This is especially important for people who are disproportionately burdened by COVID-19, such as African Americans, Hispanics, Latinos, and American Indians and Alaskan Natives. And these minority populations account for half of all reported cases of COVID-19 in the United States. I wanted to say a quick word about long haulers. I'm sure you've all heard about long haulers. We know that many organ systems besides the lungs can be affected by COVID-19. And while 80 to 90% of persons with COVID-19 recover and can return to normal daily activities, some patients can have symptoms that last for weeks and even months. And I have a list of symptoms here. I'm not going to read through them because I see we're getting a little bit short on time. But they're widespread and they can include every organ system in the body. So if at least 10% of patients are in the long hauler cohort, that means as of yesterday, Wednesday, January 20th, Inauguration Day, more than 40,000 people in the state of Alabama could be suffering post-COVID-19 illness. Nationwide, the number could be well over 2.5 million. So this is a huge, huge problem. UAB Health System has opened a new post-COVID treatment program. And that's for patients who continue to have symptoms 3 weeks or more after the diagnosis of COVID-19. It's directed by Dr. Turner Overton. The program functions as a referral system to guide patients to specialized care based on symptoms. The most common symptom to date for these long haulers is continued respiratory trouble. Men are more likely to have post-COVID-19 complications that require hospitalization, but it appears right now that women are more likely to suffer long-term symptoms. So research is ongoing. 3 weeks ago, a group of UAB scientists published a paper showing that there is prolonged dysregulation of the immune system in some of these long haulers. And this may be a factor in the persistence of the symptoms. And I've given the reference down there at the bottom of the page. But the reasons for optimism. I know I painted a kind of grim picture, and it is grim. But on the upside, there are currently approved vaccines that are highly effective. More vaccines are on the way, including some that may not require multiple injections, or some that don't require any injections at all. They can be taken with nose drops. The SARS-CoV-2 variants that have been detected to date do not appear to affect the efficacy. People who have recovered from COVID-19 seem to have fairly long-lasting immune response, perhaps 6 to 9 months. The length of time immunity lasts after vaccination may be on the same order of 6 to 9 months. But, and on a note of caution, new variants of SARS-CoV-2 continue to emerge. And no one knows how these new variants will affect ongoing vaccine efficacy and treatments. So I'm going to stop there and open the gallery up for questions. I hope I wasn't too much or too little. I tried to make this just about right. So I'll be happy to address any questions you have. I don't know, do you have a moderator for these questions sessions, or do I just field them?
Would anybody like to ask a question of Dr. Kyser?
I've got a bunch.
Good.
First of all, if you're doing an operation on somebody, they love to hear that it's 95% successful, and they are very optimistic. But I always had to tell people, that's fine, but if you're one of the 5%, you don't give a rat's ass about everybody that did okay, because you didn't. So if we're vaccinating people in 5% of the population, it's not going to work. Is it worthwhile looking for them, or is there a method to test whether or not your vaccination was successful?
Can't hear you, Kent.
Can you hear me now?
Yes.
Yeah.
Yes.
Okay, good. So those are great questions. So let me take them one at a time. If you're one of the 5% who do not have a robust response to the vaccine, you may still have reduced severity of disease if you were to catch COVID-19. So it does give you some protection against severe disease. As time goes on, too, as I tried to show in my slides, the number and type of treatments that are available are better and better. Also, there may be additional vaccines that have even higher efficacy. So that's another important reason why people should continue to wear masks. The degree of protection can be assessed in individuals who've gotten the vaccine by doing an assessment of your antibody levels in your blood. But we don't have the resources to do that for everyone. So I would just urge you to be optimistic, but keep wearing your mask. And eventually we'll reach that situation you've heard about called herd immunity, where there'll be enough people with high enough levels of immunity that you won't be able to contract the disease very easily.
This may be a related two questions. One is, since the people that get really sick with this are truly profoundly ill and terribly expensive to treat, is there a way to identify the people that are going to have a bad infection early on by any particular method, checking the virus or their particular situations? And related to that, is there any objective way, other than you mentioned the bizarre changes with the immune system, to recognize those people who are long haulers? Because I can foresee ads on TV. If you've had COVID-19, let us help you get your disability insurance. And so is there an objective way to tell who really has that and who doesn't?
Yeah. Okay, let me take the second question first. So that paper I referenced looking at the immune dysfunction, the immune regulation dysfunction, that may ultimately provide a way to determine who's a long hauler and who isn't. For the first part of your question, in terms of the people who suffer from chronic disease, if you've been diagnosed with diabetes, hypertension, any kind of cardiovascular disease, any kind of pulmonary disease, you should consider yourself at an enhanced risk for severe disease. There's no way to tell. Not everybody with a chronic condition will suffer from severe disease, but your odds are much, much greater if you do have a chronic disease. And frankly, when we get into our 70s and 80s, most of us have some kind of chronic disease. So just be careful, wear your masks, get vaccinated as quickly as you can, and take care of each other.
All right, I have a question. So far, the mutations that have shown up with coronavirus seems to have been ones that affected the infectious nature of the virus. That is, people get it more quickly. Is there any reason to think, though, that it might not mutate to become more deadly, somewhat like the 1918 flu epidemic initially was not as deadly, and then it mutated to become very deadly?
Yeah. So let me say two things about that. One, so right now there are people doing computer modeling of the viral mutations, looking for exactly that sort of thing. What would make it either more transmissible, more benign, or on the other hand, more fatal? Now, trying to compare to the 1918 influenza pandemic is a difficult thing to do, because if you think about it, the treatments then were much less sophisticated than we have now. If we didn't have the medical treatments we have now, it might be that COVID-19 would be just as deadly as the 1918 flu. We don't know, right? So it could be just that our long-term investments in biomedical research and in our health systems are the reason it's not as deadly. But yeah, you're right. There's no way we can predict which way the virus is going to go in terms of mutations. We've already seen mutations that increase its transmissibility, but so far none of the mutations seem to affect either its lethality or its or the effectiveness of the vaccines.
I have a question. Doctor, thank you for your presentation this morning.
Oh, it's my pleasure.
All of those volunteers who participated in the final trials for the Moderna vaccine and for the Pfizer vaccine, I assume they're being tracked to see the length of time for the efficacy.
Oh, you bet they are.
And that may give us more answers as to the length of time of the efficacy. Can you tell us when the final decision was made, that how long ago the final trial was approved, so we can use that as a.
So the vaccines have only been in widespread use for a couple of months. So I'm guessing by this time next year, we'll have a very, very good idea of whether the immunity from the vaccines is durable. That's a fancy way of saying that it lasts long. Or it may be that it lasts a year and then we have to get a booster like the influenza shot.
Mm-hmm.
But I feel certain within a year we'll have a much better idea of that. I'm happy to come back in a year and talk about this again if you'd like.
I have a question.
Thank you.
Ron Binnick. My question is related to the uniformity of treatments throughout the country. Is there some measurement of success so that the knowledge of what works and doesn't work is disseminated, so you don't have individual doctors based on their limited knowledge or maybe prejudice using one treatment as against another?
Yeah, boy, yeah. Yes, absolutely. So there's a nationwide network of major medical centers that are sharing best practices in terms of treatments, preventive measures, supportive measures, things of that nature. That said, I'm afraid that somebody who goes into the hospital in Scottsboro, Alabama, may not receive the same level of treatment and supportive care as someone who comes to UAB. And that's not a rap on them. They're doing the best they can with the facilities and the resources they have. It's just the major medical centers are better equipped to deal with this. But this is the best practices and methods are being shared with every hospital and every medical center in the country. But your chances are better if you can be treated at a major medical center, as a rule. And again, I don't mean that to insult anybody, any other medical center. It's just, the luck of the draw. If you live in rural Alaska, you're not going to have access to the same level of treatment. Great questions.
Follow up. If you are the individual doctor, do you defer treatment that's gone through a double-blind randomized study or something that you've heard 3 or 4 cases because it's life or death and you offer it to the patient and he says, "Doc, try whatever you know." So would you defer to double-blind randomized studies in that context?
You're putting me on the spot here.
But it is.
Yeah, so that's a great question. For new and emerging treatments that have not yet had wide-scale trials, you give it a try. I think that's up to the doctor and the patient. The doctor has to bear in mind what's ethical. First and foremost, do no harm. That's a really hard call. And I wouldn't presume to tell either a doctor or a patient the course of action that they should take.
Thank
I
you.
have a couple of questions, Mom. Ron Kogan, and they're unrelated questions. One is, why is it that the messenger RNA vaccines require 2 injections for the most part, whereas the attenuated virus vaccines require only 1 injection? My second question is about the fact that the disease is more severe in men usually, and that the long haulers are usually women. Isn't it possible that men delay seeking treatment until it's later in the disease, so that they are ending up with a more severe disease at the end because they delay treatment or diagnosis? The second part of the question is, are women more likely to be long haulers because they are usually more in tune with their physical well-being and generally notice symptoms much better than men do, and are willing, whereas the men are willing to live with them, the women aren't?
That's a lot of questions here. I'm just making a note here so I don't lose track. Your first question, why does it require 2 injections? When you use an attenuated virus vaccine, this is pretty much the whole virus. So there are lots of pieces of the virus the immune system can recognize and attack. With the mRNA virus, we're only encoding a sequence of amino acids that make up the spike protein, part of the spike protein. So that's the immune system only sees that part of the virus. So to really hammer the lesson home, you have to have a booster. That's the first answer to that. Why are men's cases more severe? It could be what you suggested, that they delay seeking treatment because men are, hardy and macho and all that stuff. It also could be that men suffer from higher levels of heart disease and obesity and other pre-existing conditions that can mean they have more severe disease. In terms of why women might be more long haulers, what you suggest could be the case. I don't know. I just don't know. I don't think anybody knows right now. And I don't really have a guess as to why women would be the long haulers. Your idea that they're more in tune with their bodies could very well be part of it. These are fabulous questions y'all are asking. I'm bowled over. Right?
I've got a question. Dottie Goth here.
Yes.
Back to the vaccines. Obviously, the 2 vaccines we've got now are producing and will lead to antibodies against the spike protein. Will from your previous remarks, should I take it that the attenuated virus vaccines that are coming may lead to other types of antibodies, which would make it more difficult for a virus to mute itself free of the vaccine?
Yeah.
Or are all of them eventually going, or are the most protective antibodies those against the spike?
Well, in terms of the degree of protection so far, the mRNA viruses are better than anything we've ever seen before. I mean, it's a remarkable level of protection. But your point is well taken. For the attenuated virus vaccines, since the immune system is seeing different parts of the virus, will it be harder for the virus to mutate and therefore evade the protection? It's a good question, and I don't know the answer.
We seem to be able to come up with new vaccines for the flu every year. Yep. As the virus mutates.
Right.
Will we be able to do that as easily with this one if it eventually mutates?
Well, the fact that we were able to, so the development of the Pfizer and Moderna vaccines began sometime in the early spring last year. And the fact that they were released this quickly for widespread use is phenomenal. Normally vaccines take 5 years to develop. So this new type of vaccine is as close as I've seen in my lifetime to a miracle. It's unbelievable. And there are other forms of vaccine coming on. As I said, there's a company making one that requires a single application of nose drops that may provide very high level protection. So the advent of molecular biology and its application to vaccine production is a phenomenal change. It's a real sea change.
I have a question. This is Kathy Rostan. What's the role of polyethylene glycol and is it proven that that's what people are reacting to when they have a bad reaction?
Yeah, that's been something that's been in the press a lot. To the best of my knowledge, and I want to be careful, to the best of my knowledge, there is no evidence that adverse reactions are caused by PEG. It's been used so often in so many different drug formulations. I would be surprised if that's it, but I can't rule it out.
So is it part of the lipid capsule?
Yeah.
Or
Yeah.
that it's
Yeah,
integral?
it helps
in
to
there?
stabilize the lipid
Okay.
nanoparticle.
Okay.
I have one more real quick question, if I can.
Sure, of course.
And that is there any evidence that the severity of disease is dose related?
Meaning
So
to
I'm
the
going to
viral load?
interpret-
Yes.
Viral load, yes.
How large a dose?
Yeah,
Expected-
that's why they say, that's why you may have read that don't get in large groups for long periods of time. Because if there's somebody positive, you could be exposed to virus and end up with a heavier initial viral load. While if you're exposed to someone for a very short period of time, you might get a very light viral load that your innate immunity could handle.
I have another question, which is not really in your field. Is there any explanation why Alabama's response to the vaccine administration is 1.9% as opposed to 3.4% for the US overall, especially when we have a center like UAB sponsoring research like you're doing?
Right. So first of all, Alabama did not receive nearly as many doses as we were initially told we would receive. When we did receive the vaccine, UAB abided as appropriate by state public health department guidelines, distributed it to other sites around the state. So for whatever reason, primarily the reduced amount of vaccine we received, plus the need to set up the infrastructure. Remember, this vaccine requires ultra cold storage before it's delivered. So we had to make sure that we had the infrastructure in place to keep the virus viable, the vaccine viable, before it was shipped to these other sites around the state. So it was a combination of things.
Thank you.
Mm-hmm.
I have a question. If the anti-inflammatory effect of metformin is beneficial to diabetics who are already on it, how does that differ from the anti-inflammatory effects of steroids, which have to be more timely and shouldn't be given early?
Yeah. That's one of the great unanswered questions. That's something that is under study right now. It turns out the mechanism, the anti-inflammatory mechanism of metformin differs from that of some of the steroids. And that's the best I can tell you. But right now, that question you asked is under study right now.
One other general question. I have no clue on how long vaccines work. I was always deliriously happy that you could get a smallpox vaccine once. And the polio vaccine came along after my time. I don't even know if I had it, but I thought vaccines were forever.
Yeah. Yeah, as the flu virus has shown us, some viruses can change so much in a year that you need a new vaccine. I wanted to say just to show you an example of another vaccine that many of you may have had that required 2 shots, the Shingrix.
Yeah.
Right, again, because that vaccine is against a limited portion of the virus. So, but only after 2 for Shingrix, you're done, and it seems to give you very, very durable long-term immunity.
On the mutations of the virus so far, are they in the DNA within the virus or on the spike proteins?
Yeah. So the virus, the SARS-CoV-2 it's an RNA virus to begin with. So and the mRNA encodes a whole bunch of things. So when the virus merges with a human cell, the virus's RNA is injected into the human cell, where it takes over the cell's biosynthetic machinery. And the virus's RNA makes viral RNA polymerase, which is the thing that makes more copies of the virus. And that makes more copies of the spike. In fact, it makes whole new copies of the whole virus. And then those viruses are transported to the surface of the infected cell, i.e. a cell in your lung. And then it pops out of the cell to infect other cells. That's how the viral infection spreads throughout the body.
Dr. Kyser, Don Long, thank you very much for your presentation. My wife and I are both UAB patients. We made application for a UAB vaccine. Are you familiar with the UAB vaccination effort? There's been some current information on behalf of UAB that suggests that UAB may be delaying vaccinations because of inadequate access to supplies of the vaccine.
Unfortunately, I think that's the case for the entire state. You know, it seems as if Alabama often gets the fuzzy end of the lollipop. In this case, by receiving inadequate supplies of vaccine, I think, but we're not alone. The federal government did not do a good job of ensuring that there were adequate supplies of vaccine and then distributing those vaccines to the states. They just really didn't do an adequately, they did a terrible job. It's frankly, in my view, and I don't mean this to be political at all, it's an embarrassment to the country.
I have one more question. Is there any explanation of why these viruses, the flu viruses and others, seems to start in the Far East and then migrate into the Southern Hemisphere and Western Hemispheres?
Yeah. So many of these viruses originate in various parts of Asia. And in various parts of Asia, many people have much more direct contact, both with domestic animals, but also with wild animals. And that contact comes about through markets in many Asian countries. Wild animals are harvested and sold as food, or they're sold because parts of them are considered medicinal or have other properties. And because there's a huge reservoir of viruses out there in the world that we don't know anything about, occasionally, and it's remarkable actually that doesn't happen more often, one of them crosses the species barrier somehow, often through an intermediary. In MERS, it was thought to be camels. In SARS, it was thought to be civet cats. And I'm sure you've all heard in this case, it may be bats. So these wild animals that are being sold for food or or other uses in Asian countries serve as an intermediary host, and the virus can jump species.
Are there any further questions? Well, Dr. Kyser, we've certainly enjoyed having you come and speak to us this morning, and we'll look forward to maybe hearing from you again, if not next year, whenever it is you can come back.
Bill, I would ask if Dr. Kyser, if you would tell us something, just a brief overview of some of the other research programs that are going on at UAB. We may want to have you back again to talk about something entirely different.
Yeah, I'd be happy to do that. You know, there are very, very strong programs at UAB in cancer treatment, cancer prevention. Mm-hmm. There are excellent programs in Parkinson's disease and other dementias. We have an Alzheimer's center as well. We have the diabetes, comprehensive diabetes center that I talked about today. Mm-hmm. We have the Vision Science Research Center where people are doing research to help prevent blinding diseases like glaucoma. We have the Minority Health and Disparities Research Center, which is trying to reduce disparities in disease prevalence and outcome across the state. There's the Free Radical Center, which looks at how certain kinds of highly reactive oxygen species can cause tissue damage. So I'm in my job in the Vice President's office, the center program is part of my portfolio. So I know a lot of these centers and center directors really well. And I know a lot about what they're doing. So I'd be happy to come back and bring some of them with me if you'd like, and we can have a session about some of the, a deep dive into some of the center programs in these areas. There are about 23 centers right now that have specialties that I sort of oversee.
Thank you once again. For those of you who'd like to join us tomorrow for Friday Forum, we'll be starting tomorrow morning at 9:30, I believe. And thank you for all joining us this morning.
I've enjoyed being with you all. Thank you so much.
Thank you. It's great.
Excellent.